So our next topic is Golgi complex. Okay. So Golgi complex is also very important cell organelle. It was discovered by Camillo Golgi. It was discovered by Camillo Golgi. It was discovered by Camillo Golgi. Okay, Camillo Golgi. So Golgi complex was discovered by Camillo Golgi. Now, if you see what is the importance of Golgi complex, what is the function of the Golgi complex? So, if you see the cell, this is considered this is a cell. So, in this cell we have nucleus. Okay, in the middle. And after the nucleus you will have your endoplasmic reticulum. So, like this, if you draw. So, this is our endoplasmic reticulum. So, this is our nucleus. This is our endoplasmic reticulum. So, that we have discussed earlier. After the endoplasmic reticulum, you have the Golgi complex situated. Okay, the Golgi complex are situated after the endoplasmic reticulum. Okay. So here, like this, so here the Golgi complex are situated after the endoplasmic reticulum, in between the plasma membrane and endoplasmic reticulum. So Golgi complex is situated in between the plasma membrane and endoplasmic reticulum. Okay. Now, what is the function of Golgi complex? Why they are important? Why we are studying about Golgi complex? Golgi complexes are important because whatever the protein is synthesized from the endoplasmic reticulum, we know that endoplasmic reticulum contains ribosomes. So ribosomes function is synthesis of proteins. So proteins are synthesized on the endoplasmic reticulum. That proteins are taken by are given. So if this is so I will draw here. So if this is our endoplasmic reticulum. So the proteins will be synthesized. The proteins will be synthesized and they are given to the Golgi complex. Suppose this is our protein molecule. Okay. Now this is our endoplasmic reticulum. Okay, rough endoplasmic reticulum. Okay, this is our rough endoplasmic reticulum. Okay, RER. Okay. On this the proteins are synthesized. Now the proteins are given to the Golgi complex. The proteins are given to the Golgi complex. The Golgi complex takes the proteins and what it does, it will modify it and pack it. Okay. Suppose if you are you are preparing some chips. So suppose a company is preparing chips. How does it sell? It will not sell loosely, no? It will pack it, it will add some spices and make it tastier and it will pack it, then it will sell it. Similarly, what the protein is manufactured by the endoplasmic reticulum or by the ribosomes present on the endoplasm. The protein is taken by the Golgi complex and it will modify it. It will either it will add some compounds to make it active or it will delete some compounds to make it active. So something, it will modify the protein. Okay, it will modify it and it will pack it. Okay, so modification. So what is the function of the Golgi complex? Modification and packing. Okay, modification and packing. So it will modify it. Why the proteins are modified? To become active or for various reasons, mainly for activation, for activating the proteins. So modified, or sometimes some like you have glycoproteins, lipoproteins. So what are glycoproteins? To the protein molecule, carbohydrate is added. That becomes a glycoprotein. Glyco means carbohydrate. So to a protein molecule, carbohydrates are added. That is glycoproteins. Lipoproteins. To a protein molecule, lipids are added. Lipoprotein. So several proteins are there in the body. So we don't need to discuss now. So that proteins are modified. So glycogen, like carbohydrate is added. So that modification is done in the Golgi complex. Now after modification, they are packed in a sac-like structures, and they are released into the cytoplasm. These are the proteins. They are packed into a sac-like structure, and these sac-like structures are called as vesicles. Okay, these sac-like structures are called as vesicles. These vesicles are released into the cytoplasm, and wherever the protein is needed, the vesicles go there and give to that particular cell organelle. Okay, that is. Now if you see the structure in detail, Golgi complex is also made up of two membranes. Golgi complex is made up of two membranes. It is present immediately after the endoplasmic reticulum. It is present immediately after endoplasmic reticulum. So whether it may be not only proteins, lipids are also modified and packed here in the Golgi complex. Not only proteins, lipids are also modified and modified and packed in the Golgi complex. So immediately after, that's why whether it may be rough or smooth, endoplasmic reticulum, after endoplasmic reticulum, Golgi complex is present. Now the third function of, if you see here, we are seeing this finger-like structures. So these are called as cisternae. So these are called as, it is made up of two membranes, outer membrane and inner membrane. So these finger-like structures are called as cisternae. Okay, cisternae. These cisternae are arranged parallel to each other. The cisternae are arranged parallel to each other. Okay, the cisternae are arranged parallel to each other. Okay. Now, from the endoplasmic reticulum, the protein reaches to the cisternae. From this, here the modification will be done. Again, here also slight first modification, second modification, third modification. So final modification is done, and they are packed here, and then they are released into the cytoplasm in the form of vesicles. So this is the structure. Now if you see the vesicles, now another function of Golgi complex is it leads in the formation of lysosomes. Formation of lysosomes. Okay, formation of lysosomes is through Golgi complex. See, this is the vesicles. The vesicles contain some proteins. If the vesicles are containing digestive enzymes, that becomes the lysosome, because the the lysosomes contains hydrolytic enzymes or digestive enzymes, whose pH is 5. So if the vesicle is packed with a protein, with the enzymes, hydrolytic enzymes, that becomes the lysosome. That's why lysosomes are formed from the Golgi complex. Next, complex carbohydrates are converted into simpler carbohydrates. Complex carbohydrates are converted, or complex carbohydrates are made from simple one. simple carbohydrates. Okay. So either it may be carbohydrate, protein, or anything, lipid. So the protein is coming or the carbohydrate is coming, lipid is coming. So they are modified. Modification means they are made complex or they are made activated and they are released into the cytoplasm. Now that the vesicles are there with the proteins or enzymes inside, they will reach, if the plasma membrane needs the protein. So these vesicles are taken to the plasma membrane and they break up and the proteins are released and these proteins are taken by the plasma membrane and it manufactures its own, whatever is needed for it, it will manufacture its proteins. Plasma membrane will manufacture its protein. So this is about Golgi complex. So just remember that Golgi complex is a very important cell organelle which is present just backside of the endoplasmic reticulum. It has finger-like structures which are called as cisternae. They are arranged parallel to each other. So what is the main function of Golgi complex is modification and packing of biomolecules, or it may be protein or lipid or carbohydrate. So they are protein, if you see endoplasmic reticulum, proteins and lipids are synthesized. So they are given to the Golgi complex. They are modified and they are packed and they are released into the cytoplasm and in the form of vesicles. These vesicles go and wherever the protein or lipid is needed, it will break and the protein and the lipid molecules are taken by that particular cell organelle. Okay, thank you.